Imagine stumbling upon a real-life example of evolution in action, where the passage of time is visibly etched on the surface of a delicious wheel of cheese. This is precisely what happened to Benjamin Wolfe, an associate professor of biology, and his team when they discovered a fascinating transformation in the fungi growing on a special type of cheese called Bayley Hazen Blue. The story begins in 2016, when Wolfe embarked on a journey to Jasper Hill Farm in Vermont with his former postdoc advisor, who had an ulterior motive: to propose to his boyfriend at the spot where they first met. Wolfe, known for his penchant for holding on to samples, ended up storing the cheese in his lab's freezer, unaware that it would later become a key to unlocking the secrets of rapid evolutionary change. Fast forward to when graduate student Nicholas Lau collected recent samples of Bayley Hazen Blue from the Jasper Hill Caves, only to find that the cheese had undergone a dramatic transformation. Its previously green, fungus-covered exterior had turned a chalky white. This unexpected discovery sparked excitement among the team, as they realized they might be witnessing evolution in action. But here's where it gets interesting. The team's findings, published in Current Biology, reveal that the color change was not just a simple adaptation, but rather the result of a complex interplay of genetic mutations. So what drove this rapid evolutionary change? The answer lies in the disruption of a gene called ALB1, which is responsible for producing melanin, the pigment that gives the fungi its green color. When the fungi found themselves in the dark, damp environment of the cheese cave, they no longer needed the protective effects of melanin, and thus the gene was effectively switched off. This process, known as relaxed selection, is a common occurrence in organisms that adapt to dark environments, from blind fish to salamanders. But what's remarkable about this discovery is that it wasn't just one mutation that caused the color shift, but rather a multitude of independent mutations that occurred across different locations in the genome. Some fungi had point mutations, while others had large insertions of DNA caused by transposable elements, which effectively knocked out the ALB1 gene. This finding has significant implications for our understanding of how fungi adapt to different environments, which is crucial for addressing issues like food security and health. For instance, did you know that around 20% of staple crops are lost to fungal rot, and an additional 20% are lost post-harvest due to mold? Understanding how to control this problem while preventing fungal adaptation is an agricultural priority. The Wolfe Lab's discovery also has potential applications in the development of new types of cheese with improved aesthetics, taste, and texture. By inoculating fresh brie cheese with the novel white mold, they created a cheese that is slightly nuttier and less funky, according to Lowe. But here's the part that might spark some controversy. Could this discovery be used to develop more resilient, disease-resistant crops, or might it have unintended consequences on the environment? And this is the part that most people miss: the potential for domesticating fungi to create new genetic diversity, which could be a game-changer for the cheese-making industry. As Wolfe notes, seeing wild molds evolve right before our eyes, over a period of a few years, helps us think that we can develop a robust domestication process. So, what do you think? Can the discovery of genetic mutations in cheese cave fungi revolutionize the way we approach food security, health, and cheesemaking? Share your thoughts in the comments below. The study's findings, published in Current Biology, provide a fascinating glimpse into the world of evolutionary biology, and we can't help but wonder what other secrets the natural world holds. As we continue to explore and understand the complex mechanisms that drive rapid evolutionary change, we may uncover even more surprising discoveries that challenge our assumptions and spark new debates. The question is, are we ready to embrace the possibilities and controversies that come with this new knowledge?